Hey guys, thought I would do a Pencast about DNA. You've heard of DNA. We've talked a little bit about DNA, but let's get some of the particulars down. DNA is actually a shortened version of the word deoxyribonucleic acid. So there's the D, there's the N, there's the A. It is a nucleic acid. And a nucleic acid is a polymer. If you remember back from first semester, we had polymers like carbohydrates and lipids and proteins. And this would have been in that chapter, but I thought it would be best saved to do in the DNA chapter. There's actually two kinds of nucleic acids, or two categories, DNA being one of them, and RNA. We're going to look at the difference between those two when we get to another process. It turns out that DNA is composed of nucleotides, and a nucleotide's a D. Nucleotide is actually itself composed of three parts. These three parts are called a sugar, a phosphate, and what we call a nitrogen base. The sugar is called deoxyribose. The phosphate group is simply a collection of one phosphorus atom and four hydrogen atoms. And we saw this phosphate group back when we talked about ATP. We actually saw three phosphate groups. And then a nitrogen base can be either cytosine, which we abbreviate C, guanine, which we abbreviate G, thymine, which we abbreviate T, and adenine, which we abbreviate A. And as a side note, adenine actually makes up the A in the ATP. So DNA and ATP have some commonalities. Well, these nucleotides, or these monomers, the building blocks, remember a monomer is a building block, are arranged in a way that most people would recognize if they saw a structure, a model of DNA. And that structure is called a double helix. So I'm going to try to draw a double helix. And it turns out that a double helix is basically two backbones, the two curving lines I just drew, with chemicals that serve as cross braces across the molecule. So this doesn't really do it justice on paper because it's two-dimensional. If we were to untwist this double helix, by the way, that's a double helix. And a helix is basically a spiral. Think of a slinky. So this would be two slinkies side by side spiraling, and these chemicals across the two sides would be basically holding the two slinkies together. If we were to untwist this helix, it would resemble a ladder. So I'm going to keep this sideways just to make sure I don't lose anybody. So by untwisting this double helix, I get a sideways ladder. Okay, so it turns out that the arrangement of all of these parts here happens in a particular way. I'm going to draw below this sideways ladder. I'm going to use the letters P for phosphate, S for sugar, and then I'll use B for base. Well, actually, I'll use one of these four letters for the bases. So if we look at this arrangement on this backbone right here, if we look at this backbone, it would appear to look something like this. A phosphate bonded to a sugar, bonded to a phosphate, bonded to a sugar, and this pattern never changes. It's what I call the boring part of DNA because there's no variation in it. And on the other side of the ladder, you have the same thing going on. You have a phosphate bonded to a sugar, etc. Now, I'm going to stop about right there. Now, what makes up these cross braces? Well, connected to the sugars are one of these four bases, C, G, A, or T. And they do that, they bond, two bases bond together to make a step of the ladder. So, for example, here might be a G bonded to that sugar, and down here might be a C. Now, C and G is what we call a complementary base pair. So this is a complementary base pair. And A and T is also a complementary base pair. And those can go in any order, C and G, G and C, A and T, T and A. But you'll never find A bonding with G. You'll never see C bonding with T. It's always got to be these two together and these two together. And what's really important to note up here is that the bases are bonded to the sugars and not the phosphates. Now, this wouldn't be a very good ladder if we didn't have something right here to hold those two bases together. So there is another kind of bond, which I'm just going to put some dots. This is called a hydrogen bond. And we studied hydrogen bonds in first semester. When we studied covalent and ionic, hydrogen bonds are actually bonds between molecules. And they're very, very weak. And we'll talk about how that's an important factor in another Pencast. So notice that the the rungs of the ladder are just bonded to the sugars. So here, let's put an A and a T in a hydrogen bond. Let's put a C and a G in a hydrogen bond. Let's extend this one more set. Let's just go all the way. Excuse me. And let's put a T and an A. Okay. And a G and a C. Excuse me. Now, if I were to put these letters up here on our double helix model, you might see a sugar there, a phosphate there, a sugar there, a phosphate there, a sugar there, etc. And you would see maybe a G here and a C there. Maybe an A there and a T there. So the bases, the nitrogen bases bond to the sugars and not the phosphates. And the backbones of the double helix model is actually a sugar, alternating sugar phosphate arrangement. So that's the structure of DNA.